So good afternoon, everyone. Welcome to the third lecture of our mini-school on quantum biology. And again, our guest speaker is Betony, that you all know by now. And Betony, I think I don't need to reintroduce you for the third time. So let's save time, and you're more than welcome to start. There was a little request in the chat a minute ago to do a quick recap of the previous lecture, because we had a little challenge with uploading video this week, and the second lecture is not yet on the YouTube channel, but should be rectified soon. Okay, Betony, thank you very much. Over to you.
Okay, thanks, Francesco, and hello, everybody. Thank you so much for coming to this third lecture of the mini-school on quantum biology. So the last two lectures, we went over some basic ideas about physics and biology of quantum biology, and we looked at some of the specific biological functions that could use quantum effects, such as coherence, entanglement, spin chemistry, and tunneling. And that was a very general introduction, so all the various biological contexts that these quantum effects might occur in. And today I want to go into more detail for just one example, and to outline the process of going about solving a basic quantum biology problem. And the example I want to look at is an example of spin biology, specifically whether quantum effects play a role in how nerves and the brain works, because it's something that I'm especially interested in. I've also tried to make this a slightly shorter lecture to leave some good time for questions. Before I move on to the specific example, I wanted to give you some context of the study of quantum effects in the brain, and the different ways in which this might manifest, both from the physics point of view and the biology point of view. So maybe the most famous and most contentious theory of quantum effects in the brain, or quantum effects specifically in consciousness, is called orchestrated objective reduction, otherwise known as ORC-OR, and it was developed by the Nobel Prize-winning physicist Roger Penrose and the anesthesiologist Stuart Hameroff in this 1989 book called The Emperor's New Mind, concerning computers and the laws of physics. Penrose suggests the possibility that the laws of classical physics aren't sufficient to explain the phenomenon of consciousness. So he suggested that quantum physics might be integral to understanding consciousness. And his hypothesis was very theoretical to start off with. It lacked a biological, because he was a physicist, it lacked a biological context in which these quantum effects might occur. And then along came Hameroff, an anesthesiologist by training, and he'd been previously interested in microtubules, and he suggested that they could be a contender in which to situate this theoretical quantum model for consciousness. And together, Hameroff and Penrose have subsequently collaborated on developing and refining this theory, but it still remains quite contentious and hasn't yet had any real experimental verification. So you heard a lot about microtubules last week, but in case you missed it or the video wasn't clear, what are microtubules? So microtubules are formed by the polymerization of tubulin dimers. Tubulin dimers consist of alpha and beta tubulin proteins, and the tubulin dimers form into longitudinal protofilaments, and then these protofilaments form microtubules. Microtubules are part of the cytoskeleton of cells. They contribute to cellular shape and structure, and they have a number of other really important functions. One of the interesting functions they have is they play cell division, and they form what's called the apparatus oversees the division of chromosomes into new divided cells. Microtubules are also a transport system. They act as motor proteins move their cellular constituents. And to remind you why microtubules are of interest in quantum biology, I need to remind you about what tryptophan is. Tryptophan is a chromophore, like chlorophyll and retinal. It is a molecule that can absorb and transmit light energy, and tubulin proteins have a number of these tryptophan molecules in them. And so these microtubules consist of these underlying networks of chromophores that might support coherent energy transfer. And if you were in the lecture last week, you might remember that coherent energy transfer is the transfer of energy between these chromophores that delocalizes the energy over possible transfer pathways, so that more than one chromophore shares the energy, rather than the energy hopping from one chromophore to another at a time. So what is interesting about microtubules in the context of quantum effects in the brain in particular is that anesthetics appear to disrupt coherent energy transfer in microtubules. And you may recognize this slide from last week. And this showed theoretical and experimental approaches to whether these repeating arrangements of tubulin dimers with their tryptophan architectures in microtubules might transfer excitation energy along the microtubule like a natural or artificial light harvesting system. And the paper on the right used tryptophan autofluorescence lifetimes to look at this energy hopping between tryptophans in tubulin and in microtubules. And they showed that this electronic energy can diffuse over 6.6 nanometers in microtubules, and that this energy transport cannot be explained by conventional Forster theory. But more interestingly and more relevantly for today's lecture is that they showed that the presence of certain anesthetics reduced this exciton diffusion. So why are anesthetics important to the study of the brain and study of consciousness? Well, as Luca Turin, who is a biophysicist, once notably put it, the one thing that we know about consciousness is that it is soluble in chloroform. Chloroform is an anesthetic, and as we know, anesthetics have a profound effect on consciousness. Anesthetics are also really fascinating from the scientific point of view, because there's such a range of molecules that can have this effect, and molecules that don't seem to have much in common at all. So, for example, xenon is a noble gas, and it's a very simple molecule compared to alfaxalone. So what is this underlying mechanism that can do something as profound as knocking out consciousness? Xenon also offers an interesting case study because, as the argument goes, xenon has no distinctive shape or chemistry, but it does have physics. So in 1989, IBM did this experiment where they used a single xenon atoms and a scanning tunneling microscope to write IBM in single atoms. And Luca Turin, this biophysicist, he suggested that you use this experiment as an inspiration to suggest that xenon extends the highest occupied orbitals of proteins and facilitates electron transfer. And to test this, he looked at fruit flies under general anesthetics, and he tested to see whether he could find an increase in spin using electron spin resonance. And the reason he did this was that he was looking for increased free electrons to prove that xenon has electronic effects in biological systems. He did find an increase in spin, but the origin of this signal is still unclear, even all these years later. And he's also starved the flies of oxygen. So there were all these mitigating factors that kind of muddied up the experimental waters. But on the topic of xenon, it has inspired another really interesting experiment. And it turns out that xenon isotopes have different effects. And xenon has a number of stable isotopes. Most of these have spin 0. And so to recap what an isotope is, it's the same element, but you've added in a neutron. So you have changed the spin of the nucleus. So it turns out that xenon has a number of stable isotopes. Most of these are spin 0, but 2 have non-zero spin. I think 3 over 2. And it's, as I've mentioned, xenon's used as an anesthetic, but when it was given to mice, the xenon isotopes with non-zero spin were a less effective anesthetic, meaning the mice didn't lose consciousness as completely. And I think this is completely fascinating because this is a extremely tiny change, and yet it has a fairly profound biological implication. Before we look at more about spin biology and isotope effects, a final word on quantum effects and brains more generally. So I've talked about how important light is in both quantum theory as well as quantum biology, and it turns out that light is very important in brain function and mental health. I've talked a little bit about neurotransmitters and nerves last week. Neural communication is mediated by the release of neurotransmitters. So when an action potential is propagating along a neuron and it reaches the axon terminal, it triggers the release of neurotransmitters into the synapse. And these neurotransmitters then move across the synapse, bind to special receptors on the adjacent neuron, open ion channels, go into the adjacent ions move into the adjacent nerve and change the drag potential and possibly cause that nerve. So neurotransmitters are fundamental to how nerves pass signals along. One important neurotransmitter I already mentioned last week is serotonin. You remember last week I mentioned it in the context of how some people have tried to apply vibration-assisted tunneling to the action of neurotransmitters such as serotonin. But serotonin plays a very important role in mood. The most common forms of antidepressant pills, such as Prozac, which I'm sure all of you have heard of, target serotonin receptors. And one of my collected biology facts that I find really interesting is that recently biologists were concerned because a species of shrimp that normally evades predators by hiding in the dark parts of the sea were suddenly swimming towards the light and being eaten. And it turns out that antidepressants from human waste had gotten into the water system and collected in high amounts. And the shrimp were eating these ingesting these antidepressants. And the main precursor of serotonin is actually tryptophan, which you've heard a lot about. And tryptophan, as I've already mentioned, is light sensitive, which is why these shrimp were seeking out the light. And I found this really amazing that something as complex as brain function and consciousness involves molecules that mediate something as simple as light responsiveness. So it seems that molecules fundamental to our state of mind are sensitive to light, but do brains produce their own light? And it turns out that all biological tissue produces very weak light across the visible spectrum, which is often referred to as biophotons, which I showed you last week, and I showed you this slide. And it turns out that brains produce their own light, and that the different colors of the light emitted are a good indicator of intelligence, or however we measure intelligence. But brain slices from different species, so ranging from a toad, mouse, chicken, pig, monkey, and human, when they were excited by another neurotransmitter called glutamate, they gave off biophotons that were redshifted for the more intelligent species, which I think is also a fascinating fact. But moving on to our specific example for today of quantum biology and the nerves and brain. Last week I looked at a variety of possible quantum effects and their biological context. It's also interesting to think about how progress made in quantum biology can be more generally applied to quantum biology more generally can be specifically applied to the context of nerves and neurobiology. So if you've been here from the beginning of this mini school, you might remember me comparing a nerve cell to a bacterial cell to show the fundamental similarities between the two. So for every quantum effect that we might see in a general cell, there could be a similar effect in nerve cells because they share a lot of the same machinery. So for example, with quantum coherence and energy transfer, a lot of the progress has been made in quantum biology has been made in the context of photosynthesis, but it's not specific to photosynthesis. It's more generally a question of the arrangements of these light reactive molecules called chromophores in proteins. And as I've already told you, it's suggested that this quantum coherent energy transport might also play a role in microtubules, and specifically microtubules in nerves, and that this could explain how anesthetics work. So another way in which quantum biology nerve processes is by considering other contexts in which vibrational theories of olfaction might apply. As I told you last week, olfactory receptors are receptors, and there are GPCRs in the body, including receptors that bind to neurotransmitters and control the actions of nerves. But today, I want to look at an example of spin biology, which I introduced last week. Spin biology has most often been applied to the biological function of bird migration, but it is being applied in a growing number of contexts, including the brain. So a quick refresher on last week's lecture, or if you missed it, what exactly is spin and spin pairs, and how do they play a role in biology? Well, spin chemistry first emerged in the 1960s to explain how magnetic fields can affect chemical reactions. And then not very long after this, in the 1970s, it was first suggested that spin chemistry might extend to spin biology through birds' use of radical pair compass in their migration. So very quickly, the quantum to biology process here can be summarized as the following. So for the radical pair mechanism, a photon excites an electron, which gives rise to a spin-correlated but spatially separated electron pair. This initial singlet state then undergoes conversion to triplet, singlet-triplet oscillations due to the Earth's magnetic field and the hyperfine interaction, which is the electrons' interactions with nuclei around it. This I would call the quantum step. Then different spin states dictate what chemical products you get out. This is the chemistry step. And then these different chemicals exert a specific signaling effect in a biological system, which might translate into magnetoreception, or it might translate into the effect of reactive oxygen species, or it might translate into quantum effects in the brain. So the radical pair mechanism looks at the spin states of electron pairs, but what about nuclear spin? So in many ways, nuclear spin is a better option for quantum effects because it is less sensitive to decoherence. And there's been a recent theory for quantum cognition that suggests that phosphorus nuclear spin is a better candidate for quantum effects in the brain. So why phosphorus? Nuclear spin relaxation is much slower than its electron counterpart. Phosphorus is also spin half, and spin half nuclei don't have a quadrupole moment, which means that decoherence happens not through an interaction of the nuclear spin with its surrounding electrons, but through the interaction of the nuclear spin with other nuclei, which is slower. And if you look in biology, there's not that many common biochemical elements that have spin half. There's hydrogen and phosphorus. So interestingly, phosphorus nuclear spin has made an appearance in the world of quantum computing as well. So in 1998, Bruce Kane suggested a way to build a scalable quantum computer using spin to encode information. Information is encoded into the nuclear spins of phosphorus atoms in doped silicon electronic devices. And then logical operations on individual spins are performed using externally applied fields. And then spin measurement is made using currents of spin polarized electrons. So if you follow research in quantum computing, you'll know that one of the major issues is decoherence, which I've mentioned before in the context of quantum biology. Decoherence can cause a loss of quantum effects, such as coherence and entanglement. Although spin half nuclei have long coherence times, they do interact with other nuclear spins. So how to stop this interaction? Well, in the Kane quantum computer, this is not a problem because the phosphorus nuclei are surrounded by silicon. Silicon has zero nuclear spin. But in biology, phosphate ions bind very quickly to hydrogen ions. And as I've just told you, hydrogen ions have non-zero spin. So the interaction with phosphorus nuclear spin leads very quickly to decoherence. And the argument goes that in order to preserve the spin coherence, phosphates need to bind to spin zero ions, such as calcium and oxygen, which results in calcium phosphate. And calcium phosphate is very prevalent in biology. You might know it as the main material that bone is made out of. Calcium phosphate can take a number of forms in the body, but for the purposes of this talk, I want to specifically mention one of these forms of this is called the Posner cluster or molecule. And very simply, a Posner molecule is just a very specific type of amorphous calcium phosphate, which takes a flattened cube. This cube has 8 calcium ions on the corners of the cube, 1 calcium ion at the center, and on the 6 faces of the cube are phosphates. And phosphates are made up of phosphorus ion bound to oxygens. So why is amorphous calcium phosphate interesting? Well, for one thing, it's an important reservoir of free calcium and phosphate ions in the body, both of which are really important for a number of biological function. And recent research images mitochondrial reservoirs of amorphous calcium phosphate granules, pointed out by the yellow arrows. There's been a lot of research on the impact that stresses such as chemicals can have on the level of free calcium that comes out of these reservoirs of calcium phosphate. But there's been very little research regarding how magnetic fields can have an impact on these calcium phosphate reservoirs and the balance of calcium phosphate molecules and free calcium. Because remember, calcium phosphate, that's the calcium is bound into the, so it's not free calcium. If those molecules dissolve, then you're getting free calcium ions out of that. And why is the ratio of free calcium ions important in the context of the brain? Well, calcium ions play a critical role in the functioning of the nervous system, particularly in the process of nerve firing and neurotransmission, which I've already talked about, but we'll explain in more detail now. So when an electrical signal or action potential reaches the end of a neuron, it causes voltage-gated calcium channels in the membrane to open. And this allows calcium ions to flow into the neuron and from the extracellular space and driven by the steep concentration gradient. And the influx of this calcium triggers the fusion of neurotransmitter-containing vesicles within the cell and leading to the release of neurotransmitters into the synapse. And these chemical messengers then bind to receptors on the adjacent neuron and continue the signal transmission. So calcium is fundamental in initiating the release of neurotransmitters that then pass along a signal in nerves. And beyond just the signaling process, calcium ions are very important in brain processes, gene regulation, gene expressions, plasticity, learning, and memory. And dysregulation of calcium signaling is linked to a number of neurological disorders like Alzheimer's disease and epilepsy. So let's get to the spin biology. So a theory by physicist Matthew Fisher has outlined how entangled phosphorus nuclei in Posner molecules might act as entangled qubits, and this would result in entangled neural firing, which could then possibly change change brain function. So how do these phosphorus nuclei become entangled in the first place? So as I understand it, and there was an initial hypothesis that was then updated, but the initial hypothesis goes that suggests that the process starts with the production of adenosine triphosphate, which is ATP, in mitochondria. ATP, as I introduced in the first lecture, I think, it's like the fundamental currency of energy in the body, and it's a molecule whose energy-rich chemical bonds fuel most cellular processes. And as the name suggests, triphosphate, ATP has 3 phosphate groups, and one of the products of ATP is pyrophosphate, which is 2 bound phosphates. And the hypothesis is that pyrophosphate undergoes an enzyme catalyzed reaction to give 2 phosphate ions whose nuclei are in a spin state known as a singlet state. And it's suggested that because the enzyme stops the rotation of the pyrophosphate, due to spin considerations, it favors the production of the singlet state. So now we have 2 spatially separated phosphorus nuclei whose spins are in a singlet state, which is an entangled state. And if you remember from our discussion of entanglement, this means the spins have to be considered as a single system. They are inseparable. If then end up in different Posner molecules, then those Posner molecules might also be considered as entangled. And then this spin entanglement is then translated into a chemical outcome. Molecules bind, melt, and release calcium ions. So this is the way in which entanglement can affect Posner molecule binding and subsequent free calcium levels. So just a reminder, this isn't the radical pair. The radical pair looks at electron spin chemistry. This is the nuclear analog of the radical pair. And to summarize this model of cognition, enzyme catalyzed production of phosphates from pyrophosphates gives you a spatially separated but spin-entangled nuclear spin state. This entanglement in turn modulates chemical binding and subsequent free calcium ion production. And then finally, this concentration of calcium ions translates into a number of biological outcomes, not least of which is how nerves work and how the brain works. So up till now, I've been talking just about pure Posner molecules, by which I mean just Posner molecules that are only consisting of calcium and phosphate. So they only have calcium, phosphorus, and oxygen in them. But amorphous calcium phosphate, if it assembles in a medium that contains other ions, like body fluids, which have sodium, magnesium, and lithium in them, they can also form impure aggregates, where some of the calcium ions are replaced by other ions. For example, it is possible to have a amorphous calcium phosphate with the central calcium replaced by 2 lithium ions. The reason you need 2 lithiums for every single calcium is because calcium is divalent and lithium is monovalent. So why lithium in our problem that we want to solve? Well, from a physics point of view, we are interested in nuclear spin dynamics, and lithium has spin. Lithium also has isotopes for comparison of spin effects. From a biology point of view, lithium is important in that it's used to treat bipolar disease, which is a mental illness where periods of depression alternate with periods of mania. Bipolar disorder has been linked to neural excitability, and lithium has been shown to treat this excitability. And the Posner molecule hypothesis links spin entanglement to neural excitability through this free calcium production. So we thought it would be interesting to see how incorporating these different lithium isotopes into Posner molecules might change the entanglement and thus change free calcium ion production and change neural excitability. It's also been suggested that one way in which to probe consciousness is to understand how certain molecules, such as anesthetics, have an effect on consciousness. And while the effect is less dramatic than anesthetics, like pharmacological interventions such as antidepressants or antipsychotics, also play some role in mediating consciousness. So we investigated how lithium isotope substituted Posner molecules change quantum effects in Posner molecules. Just a word on isotopes. I've already introduced the interesting isotope effects of xenon. It turns out lithium also has interesting isotope effects. Lithium isotopes have been shown to have a different behavioral effect in rats. One experiment showed that lithium altered the parenting behavior of rats, with lithium-6 having more of an effect than lithium-7. And a more recent experiment showed that lithium isotopes have different effects in treating ketamine-induced mania in rats, with lithium-6 having more success in treating mania than lithium-7. Okay, so now we have the background context. We have the quantum effects that we're interested in and their biological context. How do we go about solving a simple quantum biology example? So if you remember in the very first lecture, I took you through the basic quantum physics concepts you would need for quantum biology, starting with what a quantum system actually is. So what is our system here? Well, we are interested in nuclei. So if you remember from my introduction of the atom, a nucleus is the dense area at the center of the atom that is made up of the charged protons or neutral neutrons, which each have a spin of a half, the same as electrons. Then the spins of all the protons and neutrons add up to give the overall spin of the nuclei. For example, phosphorus nucleus commonly has 15 protons, 16 neutrons, meaning it has one spin half left over, giving the total nucleus a spin of half. And different isotopes have different neutrons and thus different nucleus spins. So our full system that we are interested in, the problem that we're going to work through is, in this Posner molecule model, is 2 Posner molecules, each with 6 phosphorus nuclei of spin half, as well as 2 molecule. And this gives us a full system of 16 spins. And depending on whether we look at lithium-6 or 7, it will give us a different lithium spin, 6 and 3/2 for lithium-7. So if that's our system, then what state are we interested in? In lecture 1, we went through how the system is the thing you're interested in. The state is information or properties about the thing. So our system is a bunch of nuclei, and our state of interest is the spin states of these nuclei, remembering that spin defines how quantum particles behave in magnetic fields. And in particular, we will be looking at pairs of nuclei that started off in a specific state called a singlet or entangled state, but then can change over time into one of three states called triplet states. So once we have our system and state of interest, how do we go about the next step? Well, if you were in the first lecture, you will know we need something called the Hamiltonian. In quantum mechanics, the Hamiltonian is an operator that represents the total energy of the system. And the Hamiltonian also plays a central role in determining how it evolves. What does the Hamiltonian for a system of phosphorus and lithium spins in Posner molecules look like? Well, we use a Hamiltonian that includes a Zeeman term. The Zeeman term accounts for the interaction of the spin, the nuclear spin, with the external magnetic field, which is the Earth's magnetic field. It also has a J-coupling term. The J-coupling term describes the indirect spin coupling, which is the coupling between the nuclear spins in a Posner molecule. Indirect coupling, you can think of it as the, if you are more, if you know anything about the radical pair, then indirect coupling is kind of the nuclear equivalent of hyperfine coupling in the radical pair. With indirect coupling, the nuclei interact with each other, but through the electrons between them, not directly. And the strength of this coupling is given by J. So from the viewpoint of physics, we now have a model, but to make this model relevant to the biological context, we need some reasonable parameters. And this is often where the problem comes in quantum biology. So the gyromagnetic ratios for phosphorus and lithium, which will give us the Zeeman effect parameters, are well defined, but the J coupling strengths are less easily accessible. There has been some DFT modeling to calculate the J couplings for a specific Posner molecule with an S6 symmetry. This gives 3 J coupling strength, but this S6 symmetry is not guaranteed. And one of the things that different Posner molecule assemblies would do is change the J coupling values. And there doesn't seem to yet be any consensus on what these J coupling values are. And for this reason, and because it's still an open question whether these Posner molecules actually have this S6 symmetry, we thought it would be interesting to investigate how a range of different J coupling strengths have an effect on entanglement. For lithium J coupling strengths, it's difficult to find these parameters in the literature, and we had to use DFT calculations. But we do know that the relative J coupling strengths of the 2 lithium isotopes depend on their different gyromagnetic ratios. So we could work out the relative J coupling strengths for the different lithium isotopes. So we are interested in quantum effects such as entanglement and coherence. So we had to decide on appropriate ways to measure these effects. So to measure the coherence of correlated Posner molecules, we followed an approach that uses the relative entropy to measure the coherence. And this very basically gives us a distance measure of our density matrix from the maximally mixed state, which would have no coherence. For entanglement, we used concurrence as a measure of entanglement, which just tells you how much singlet you have left in your density matrix. Next up, what happens to the state of our system over time? How do we model the time evolution? This is the role of the Schrödinger equation, as you saw in lecture 1 for closed systems, or if you have an open system, which interacts with the environment, you need a master equation. Today, we're going to keep things simple and mostly only look at the coherent or closed dynamics, which is just the first term on the right of the equation. And finally, how do we solve our equation? Well, methods and Python programming, which I won't go into here, but there are really excellent options for solving spin biology problems. In particular, I'll point out mole spin, for radical pair, but is flexible enough to deal with the nuclear spin case as well. And it can analyze a bunch of interesting things about your system of choice. I'll go through some of the results. In this example, we looked at pure Posner molecules without adding lithium. and following from the suggestion that different Poisson molecule symmetries change the J coupling, we looked at what happens to the coherence, which is the top graph, and concurrence, which is the bottom graph, for different J couplings. For the coupling strengths of the order given by the literature, very little concurrence. Remember, concurrence is entanglement. Very little concurrence remains when the interactions of all of the phosphorus nuclei are considered. You can increase the amount of entanglement, though, by increasing the J coupling strength. Here we were interested in what happens to the coherence and concurrence if the Poisson molecules aren't symmetric, so they have all different J coupling strengths. We tried two different asymmetric configurations by varying the J couplings. In both cases, both coherence and concurrence are almost non-existent. In example 1, where the J couplings are different but still more symmetrically assigned, there's a tiny bit of concurrence. But if the J couplings are assigned in a way that there's a more strongly coupled half of the Poisson molecule, then the entanglement completely disappears. So this looks very confusing, but our discussion of entanglement is different to the original hypothesis, which uses the entanglement of pseudospin, which means that the full Poisson molecule is entangled rather than just one of the spins. So to try and look at how we might talk about entanglement of the Poisson molecule as a whole, we looked at whether entanglement is transferred across other phosphorus spins in the Poisson molecule. So for the symmetric case, we again looked at a variety of J coupling strengths, and each of the graphs in this grid represents the entanglement of each phosphate. So for example, phosphate 0 with phosphate 0 is the original entangled pair. And it's very clear that only the furthest phosphate inherits any of the entanglement. We were also interested in the asymmetric case. Here the blue graphs are symmetric and the orange asymmetric. And it's quickly obvious that any entanglement transfer depends on the symmetry. There's no entanglement transfer for the asymmetric case. So far, this has all been about pure Poisson molecules. Remember, when I say pure Poisson molecules, I just mean Poisson molecules with no lithium. We wanted to check whether the different spin of lithium isotopes had any effect on coherence and entanglement. We've combined this here with checking whether there is any coherence or entanglement transfer as well. The results don't look very promising, so don't be fooled by the coherence case. If you check the x-axis, only the first of the graphs is to scale. So coherence is transferred across all phosphorus nuclei, but it's almost negligible to begin with. And there's no appreciable concurrence, entanglement, both lithium isotopes. Adding in the spins of both lithium isotope pretty much destroys all the entanglement. For the case of coherence on the left, we increased the scale of the y-axis just to illustrate how coherence is transferred differently between different nuclei. And greatest coherence transfer is seen in the pure Poisson molecules, followed by lithium-6, and lithium-6 is red and lithium-7 is green. So are the lithium isotopes doing anything interesting? If we look at the spin dynamics for different spin states, you'll see that no lithium, the 3 triplet states are identical. Adding lithium separates the entangled triplet from the other triplets. It doesn't really look that good for entanglement being so robust from the results so far. And also there doesn't seem to be any difference between the lithium isotopes. Is there any way in which we might enhance or preserve entanglement, or has nature managed to do this in any way? Well, this paper looks at entanglement in radical pair magnetometers. So that's electron spins, electron radical pair. For the 2 correlated spins in a radical pair, I've already told you there are the 4 possible states, the singlet and the 3 triplets. 2 of these states are entangled. The singlet is entangled and one of the triplets is entangled. The other 2 triplets are not entangled. And this paper points out that it's possible to create an entangled subspace by using a magnetic field to separate the entangled from the non-entangled states. And this would mean applying a magnetic field that is much greater than the strength of the hyperfine coupling. So remember, this is the case of the radical pair. So it's hyperfine coupling, not J coupling. And this is an illustration taken from the radical pair literature of the high field effect. So you can see as the external field grows in strength, the 2 non-entangled triplets separated in energy from the entangled singlet and triplet state. the singlet triplet mixing goes down and the singlet product is increased. So if you could create a subspace for your radical pair that was occupied in entangled states, would this be better than having radical pairs that were in a singlet state? And is the entanglement interesting in its own right, or is it just the singlet state that is interesting? And can a similar principle be used to isolate the entangled states of nuclear spins? So now looking at this in the nuclear equivalent. So as you already saw, this is the spin Hamiltonian that we use to investigate the coherent spin dynamics of these entangled phosphorus nuclei or entangled Posner molecules. Similarly to the simple radical pair Hamiltonian, there's the Zeeman term. And in the nuclear case, you don't have the hyperfine, but you've got this J coupling term that describes the interaction with other nuclei via intervening electrons. So what happens when this J coupling is much smaller than the Zeeman coupling? Well, interestingly, it could be argued that nature supplies this entangled subspace in the spin dynamics of nuclear spins. This is because the Zeeman effect from the Earth's field is much bigger than these J coupling strengths, which I think is particularly interesting because everybody talks about how when these entangled phosphates bind with hydrogen, it's a bad thing, causing a loss of quantum effects. But because of the difference in scale of the external magnetic field, as well as the different Larmor frequencies of hydrogen and phosphorus, what actually happens is that the entangled states could be favored over the non-entangled ones. For instance, there's mixing between all states in the first graph where the Zeeman and J coupling strengths are of the same order. But if you put in the actual values, the values that nature has supplied, that's the specific J couplings of phosphorus and hydrogen and the Earth's field strength, there's this suddenly no more oscillation with the two non-entangled triplets. So in this way, binding to the hydrogen in some sense kind of preserves the entanglement. So having investigated the coherent dynamics, we were interested in adding in the dissipative or open dynamics using the master equation approach. I'm not going into much detail with this. I just wanted to introduce the idea of transition operators and frequencies because there's some interesting implications from this. The system Hamiltonian, as I've already shown you a couple of times, has the Zeeman interaction and the indirect or spin indirect or J coupling interaction between these phosphorus nuclei and the Posner molecules. If you remember from lecture 1, dissipation happens through interaction with the surrounding environment, which we can represent using this Barth Hamiltonian, where omega n is the frequency of the nth oscillator. We use a Markov approach, which if you remember means that the Barth doesn't store information about the system. And we write this master equation in terms of transition operators, which describe transitions between the levels in the system. So between the energies of the different spin states, where omega t are the associated transition frequencies. And why I find this interesting is that it can tell you about what you might call the sensitive or resonance frequencies at which the Barth might match the system. And I thought it would be instructive to compare the electron radical pair with the nuclear spin pair to see what range of resonance frequencies we are looking at. Sorry. For the electronic case, the transition frequencies are written in terms of the parameters of the Hamiltonian. So that's the Larmor frequency of an electron and the hyperfine coupling constants. And these frequencies range from kilohertz to megahertz because they depend on these hyperfine coupling constants. In the nuclear case, the transition frequencies similarly depend on the parameters of the Hamiltonian, which is again the Larmor frequency of phosphorus and the J coupling constants. What is interesting is that because of the high field effect, you will mainly get mixing that is determined by these J coupling strengths rather than the Zeeman parameters. And for nuclear, will vary for different molecules, but are mostly in the range of hertz. So why did I want to underline this comparison between the transition frequencies for the radical pair frequencies for a Posner pair? Well, in this paper, they note that the most convincing experimental test of the radical pair mechanism has involved subjecting caged birds to weak radio frequency fields, and magnetic fields oscillating at frequencies in the range 1 to 100 megahertz are expected to alter the yield of radical pair reactions by modifying spin dynamics. And this has been observed experimentally for organic radicals in solution, when the frequency of the applied field matches the singlet triplet interconversion frequency arising from these hyperfine interactions. And this is an explanation for the fact that anthropogenic noise across this broad band range of frequency disrupts birds' ability to navigate. But what does this mean for the Posner molecule or nuclear spin case? Well, for that case, the spin states interconverted frequencies given by these J couplings, which are of the order of hertz. And what I think is potentially interesting is that there's background electromagnetic radiation of similar frequencies in the body, for example, from the brain. And brain waves vary in frequency across very similar values to these J coupling constants. Exactly what this might mean, if anything, I don't know, but it could be interesting to add this noise into our master equation and see what happens to the spin dynamics of the Posner molecule. And targeting these transition frequencies, for example, stimulating conversions using resonant frequencies, might also offer a way to experimentally confirm spin effects in Posner molecules. So I'm going to bring things to a close now, but if you're interested in reading more about this, there are some papers that I showed you last week. There's far less research on this example of spin biology than on the radical pair. And I think this is partly because the radical pair mechanism is very well developed through the field of spin chemistry. But it's much less clear how this spin correlation between nuclei can give rise to different chemical outcomes. But there have been some theoretical papers looking at this topic. There's also been an interesting recent paper that looked at lithium-doped Posner molecules experimentally, and it showed that lithium isotopes differentially alter calcium phosphate mineralization, so that would be the solid state rather than the free calcium ions, in common biologically relevant solutions. And as the authors point out, this isotope effect is pretty unexpected from classical chemistry principles, but would be well predicted by something like quantum biology. It's always fun to end things on a speculative note. So here's a thought to leave you with. If our brains, and thus potentially our consciousness, does use something like spin in its function, then this would behave differently in different contexts, for example, different magnetic fields. So I've outlined how the Earth's magnetic field is the perfect magnitude to generate this entangled subspace for nuclear spin. If this field were much weaker, for instance, this wouldn't happen. You'd have equal mixing between the entangled states and the non-entangled states. This means that perhaps our brains and then maybe our consciousness would be changed if we were on planets with different magnetic fields. So perhaps it's something to keep in mind in our future efforts to become a multi-planetary species. And that's it for today. Welcome to ask questions now. And next week, we'll look at an example of electron transfer and tunneling in the mechanism by which the SARS-CoV-2 virus invades host cells.
Yeah, Betony, thank you very much again for a great lecture. Thank you very much. Do we have any questions from the participants? Any questions in this group? Any question from the participant? Please just raise your hand and we can give you the right to talk. Maybe while people think about questions, there is a question in the Q&A by Charles Johnstone, and he asked, what is the source of biophotons in the brain?
Well, I mean, that's a really good question. That because, I mean, they don't know why there's this. Let me break that into two answers. Biophotons are generally produced in metabolic processes, so, oxidative processes. That's where because they're biophotons in all living tissue. But the redshift of the biophotons in the brain of different species is not yet understood. So I think they would like to redo the experiments as well and see that it's not just some artifact, but yeah, it's still an open question. But generally, biophotons are produced in metabolic or oxidative processes.
Thank you. Any other questions or something in the chat? You have a question? Yeah, please go ahead.
Betony, back to your slide at the beginning, you mentioned something about xenon, and can you just quickly bring that slide, the xenon and the HUMO concept?
The xenon and the IBM, or
yeah. That one.
Sorry, this one.
Yeah. Okay, and it's mentioned xenon atom extend the HUMO, which leads to what? It can increase the probability of
Electron transfer, yeah.
electron transfer.
Yeah.
But do you know how these two is related?
Well, I guess the probability of the transfer depends on how the lowest. So if you have the donor of one and what the overlap of highest occupied molecular orbit and the acceptor molecule. So if you're extending it, it's going to increase that overlap, as far as I understand it.
Okay. But it's mentioned in the Luca-Turing paper.
Yeah.
I will check the paper. Thank you.
Any other comments or questions? Please, if you would like to ask the question, just raise your hand, your virtual hand, and we will give you the right to the allowance to talk.
You're also welcome to email me if you have questions, actually.
Okay, Eleanor, here comes the first question. Voila, Eleanor, you should be able to ask your question now.
Hello, thanks again, Betony, for another amazing talk. I've got a little bit of a stupid question with regards to what you said at the end. Did somebody check the effect of I don't know, NASA pilots or whatever on the brain in that sense? You know,
They have, unfortunately, though, the ISS still falls within our magnetosphere pretty much. So, and the space travel definitely has effects on psychological, psychiatric effects, but there's so many confounding factors. Yeah. But as far as magnetic fields go on the ISS, so wherever they spend an appreciable amount of time, there's still pretty much a magnetic field. Apparently, the Earth's magnetic field even extends to the moon at certain times of the year. That's why they plan lunar missions around. But you'll have to double check me on that. So it would be then going to somewhere like Mars, where there's pretty much zero magnetic field.
No.
That would have any appreciable effect.
Thank you.
Okay, thank you for the question, Eleanor. I don't see any more raised hands or questions, Betony. So, please, go ahead.
Can I ask a question, Betony?
Yeah.
Can I ask about the Posner, the entanglement in the Posner molecules? So, just if you go to the slide where you had the Hamiltonian.
Sorry, too over-enthusiastic. Yeah.
Yeah, this one. So in the simulations you did, you basically just take these operators to be, like, you basically look at a spin-half system, but these are just like the Pauli operators, right? And you have basically spin-halves. And you're just looking at...
The lithiums have different spin.
It can be spin one as well. So it's not always half.
Because I was trying to understand, like, what are you actually computing the concurrence between?
So it would start out as just one of the phosphoruses, two spin halves. So the entanglement is always between the spin half nuclei and the original ones.
Okay.
And then with the, we didn't ever look at entanglement between so it was always between phosphate nuclei, never with it was just the effect of the lithium spins on the spin dynamics, but then it was always, we traced out everything else and then just got the concurrence of the two original.
Okay, yeah. Because I was also going to ask, like, because in your plots, it looks like you have this kind of like entanglement revival. And I was going to ask, like, what is like the origin of that entanglement revival? Which I guess it's the interaction of the spins.
Yeah, it was, it was because they were interacting with these. Yeah, because we didn't even consider, we weren't losing anything to the bath. It was always, these are all closed dynamic.
Okay. Yeah, and I guess also a question maybe is similar to how you actually determine the parameters. How do you choose the initial conditions? Maybe you mentioned something about that, but I don't remember what you said. Like
Initial conditions of the simulation.
Yeah.
So we're just having two entangled, and all the rest in just thermal states.
Okay. And was that like an accurate reflection about with like?
Yeah, this is a loaded question in quantum biology. I'm not sure how accurate these assumptions are. It was more, the point was more to look at whether there was a very big difference between the two lithium isotopes on the entanglement, which actually what happens is the minute you add too many spins, everything just goes flat. So there was no difference between like entanglement between both the lithium 6 and lithium 7 just killed the entanglement.
Okay.
So it would be more. It would be more interesting to look at things like phosphates binding to hydrogens. So when there's not all these other, because they say, I mean, it seemed to me a little bit of a contradiction to say that you have this, because they phrase it as having the spin zero cage around these initial entangled phosphate. But actually, like there's a bunch of other phosphorus spins in there, spin halves that are interacting. So it's not a really isolated entangled pair. So I don't know if that answers your question.
Yeah, that does. Thanks.
Betony, there is a question in the chat by Tsepo Paul Maeka. Sorry, why is nuclear spin less sensitive to decoherence?
Because it doesn't interact with, especially for spin half nuclear, there's no quadrupolar moment, so it doesn't interact with electron spins. It only interacts with other nuclear spins.
Okay, thank you. And now the question keeps coming. Jenna Gibson asks a bio question. How would have organism evolved to utilize quantum effects? Is it a natural trait in terms of selection, or is it possibly advantageous for some things?
Well, I would have thought, so for something like spin biology, evolution would have picked the amino, so evolution works on picking the proteins that are useful. So like if you look at the radical pair, these interactions between the hyperfine interaction depends very much on what proteins you have in the vicinity of the radical. So to give you this singlet triplet outcome. So evolution could have worked by picking the right proteins to give you a certain singlet to triplet ratio. And if that singlet to triplet ratio corresponded to some useful property like magnetoreception, or in my opinion, it would have corresponded to something like reactive oxygen signaling states, then those amino acids would have been selected for by evolution. So I think there's a fairly clear mechanism. And then say for electron transfer or coherent transfer, it would be the thing that evolution could tune would be distances between chromophores. So those would be the right distances that gave you optimal coherence would be selected for. So there are quantum parameters that could be selected for, mostly around like what the proteins are, what are the distances between chromophores, what are the spins of the various nuclei that are around these radical pairs. That in that way, evolution could have acted very clearly, I think, on quantum parameters. I don't know, does that answer your question?
I think it does. Thank you, thank you very much, Betony, but I can't speak for Jenna. No, so Betony, thank you very much again for a great talk. Yeah, and Jenna confirmed she's also happy. And we will try this week not to be sloppy with the upload of videos and fix our pipeline problem that we had. So people will find all the lectures soon on our YouTube channel, and if you share the slides, we will upload them on the website as well. Yeah. So Betony, thank you very much. Then please, to all the participants, join us again next week. Yeah, same Zoom link, same time, and for those of you that are in the seminar room, same seminar room. Yeah. So Betony, thank you very much. We'll chat again tomorrow morning. Yeah. Thank you.
Thanks. Bye-bye.
Best.
Bye.
Bye-bye. Cheers. Bye-bye.